Hello, my online learners, and welcome to Chapter 10, the Muscular System. We're going to quickly start by looking at the three types of muscles and then spend the rest of the chapter focusing mainly. Okay, the three types of muscles can be determined histologically. When you look at them, it depends on the location of the nuclei, whether they have serrations or not, or striations or not, whether they're branched, and also the general shape. So with skeletal muscle, they kind of look like bundles of logs stacked. You've got your nuclei on the outside and they have these striations. When you look at cardiac muscle, it also has striations. It's got nuclei more in the middle, and it is branched. And you can also see intercalated discs, which isn't shown really well on this animation. Then the third type is smooth muscle, and smooth muscle is found in the gut. They're long, thin, flattened shapes, no striations, and the nuclei are found in the middle.
Skeletal muscles are voluntary muscles. This means you are in control and choose when to contract them. Nerve impulses from the brain trigger the muscles to contract. They attach to bones via tendons and enable movement. Skeletal muscles can produce
Skeletal muscles are voluntary muscles.
Or small
this means you are in control and choose when to contract them.
Or shorten,
Nerve impulses from the brain trigger the muscles to contract.
Fixator muscles
they attach to bones via tendons and enable movement.
Purpose is to
skeletal muscles can produce powerful, fast movements,
the rotator cuff
or small precision actions, and have the ability to stretch or shorten
Involuntary
and still return to their original shape.
Organs and in blood vessels.
Fixator muscles are a type of skeletal muscle whose primary purpose is to stabilize joints.
They contract and relax.
For example,
automatically.
the rotator cuff muscles help stabilize the shoulder.
Also known as smooth muscle because they do not
Involuntary
have the
muscles are found around organs and in blood vessels.
Smooth muscle
we do not
is found
consciously have the ability to control this type of muscle.
Esophagus,
they contract and
bronchi,
relax
and in the
automatically.
walls of blood vessels.
Involuntary muscles are also known as smooth muscle because they do not have the striped appearance that skeletal muscles have. Smooth muscle is found in the walls of hollow organs such as the stomach, esophagus, bronchi, and in the walls of blood vessels. Involuntary muscles help move food along the esophagus or constrict blood vessels to reduce blood flow.
Cardiac muscle is highly specialized and resistant
Cardiac
to
muscle is found solely in the walls of the heart. It has similarities with skeletal muscles in that it has a striped appearance.
Now, when you're looking at the detail between skeletal muscle, realize that the muscle is a large collection of many muscles. And a good example of this is if you are cooking a chicken breast. That chicken breast is a muscle, and then the fillet underneath is your pectoral minor muscle. And then if you take and you cook a muscle or cook meat slowly and with acid or heat, you will often get it to break down into muscle fibers, such as when you have pulled pork. Those muscle fibers are held together by connective tissue or fascia in order to become muscle bundles. So the active group is actually a muscle fiber, but many muscle fibers go together, sort of like fibers to make a stronger rope. So when you look at the basics of it, the basics is the muscle fiber. And the muscle fiber would be each one of these, okay? We will later on look at the myofibrils, and we're talking about something called a sarcomere. So here are the striations that are important later on. Good. Then we have a mycinium, which covers. We have a perimycin, which is an epimycin, and an endo. So endo means inside, peri means around, and epi means on top. And these are connective tissues that help hold the muscle fibers together, and they hold it all together quite strongly. Now, the muscles themselves are held together to bones by tendons. And all of this connective tissue is called a fascia, and the fascia will be outside the muscle. Often, well, if you skin a chicken, you'll see there is connective tissue or fascia there. And then you'll also see that around I'm talking raw chicken breast, there is connective tissue. So that is what we're looking at in terms of the fascia, or the connective tissue that holds them together. Notice also that inside each muscle there is a really good blood supply, arteries and veins, and then also a good nervous supply. Now, if we look at each one of the muscle fibers themselves, so this is a muscle fiber, and then the muscle fiber breaks down to filaments. Let's get the terms here. So there are the muscle fibers. The muscle fibers are long and thin, and they have many parts to them. In between each one is called the sarcomere. So when we talk about these striations are due to the different types of myofibrils inside. So let's look at the terms. There's a lot of terms, but I want you to realize they start with the word sarco, and you'll also see the prefix myo. So sarcolemma is, you may have heard of the term plasmalemma. Sarcolemma is the membrane around. Sarcoplasm is sort of like the cytoplasm, and that is inside the cell. Then you have the long bundles, and these are the myofibrils, which will fill up the sarcomere. Fill up the sarcoplasm. It's in the myofibers where there'll be stored glycogen. And if you remember back, glycogen is a complex carbohydrate of a bunch of glucose formed together in a branching pattern. Now, we talked about the endoplasmic reticulum, and we talked about the cells. In the muscles, they're specialized. They're called Sarcoplasmic Reticulum. And these are basically flattened sacs. And these are important because this is where the calcium is that you need to get the whole thing started. Calcium is a very important mineral, and it'll trigger this movement. Then we have the myofibrils. We have myosin, which is myosin, which is thick, myosin, thin, actin. Okay, and they are arranged, and that's what gives the sarcomere, the sarcomere, this striation. So here you can see the purple one are the thick ones, and then you can also see the thinner ones called actin. So it's this actin and myosin that are the filaments where contraction occurs. Now, this is really quite a complicated process. So I'm going to introduce you to the players, and then we'll look at a video to break down the process. So you have the thick myosin, and on the thick myosin are these myosin heads. And these myosin heads will grab on, bend, and pull the actin past. So the thick filaments, myosin, myosin head. Very important. Then with the thin filaments are actin. But the actin has a couple of components with it. You've got tropomyosin and troponin. Now, the tropomyosin is this rope-like protein which block the sites where the myosin will normally bond. And troponin is the molecule that will pull on the tropomyosin. So, actin, troponin, tropomyosin. Troponin is attached to the tropomyosin, and tropomyosin blocks the binding sites where the myosin normally acts. Now, when this is all set up, don't worry about the term Z lines. When it all set up, it looks like this with the actin and the myosin sort of alternated. Now, when they form cross bridges, and the myosin will join to the actin, and that will pull. But life isn't that simple. So I'll walk you through it with a video. Okay, so we are now looking at a filament of a muscle, and the sarcomere is the anchoring line for the actin. So here we have our actin molecules, and then here we have our myosin in between, and here we have our actin, and it's all anchored to the Z line. So we'll go through this video. I'll do it in steps, and I'll explain as we go because it's a very good representation.
The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move.
Okay, in basic summary, the myosin heads will grab the actin, the myosin heads pull, the actin slides.
This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP.
Okay. This is why you need energy. It's the ATP, which is the higher energy molecule, which will activate this head. Now realize that ATP, adenosine triphosphate, once it's activated, will break down to ADP, adenosine diphosphate, and phosphate.
And inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin.
Okay, so let's go through this. We'll later on in this unit, we'll talk about what happens when the nerve stimulates the muscle at what's called the neuromuscular junction. But to summarize, basically what will happen is calcium is released. So calcium is the molecule that gets this all going. So to look at the players again, we've got troponin, which is the blue balls, and the troponin is holding the tropomyosin, which is the rope. And the tropomyosin, the rope-like protein, is blocking the binding sites so the myosin can't form cross bridges. So with the stimulation of the muscle, calcium will be released.
When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin.
Okay. Now, did you see that activation? Calcium binds to the troponin, which then pulls the tropomyosin off. So now those binding sites are available so that myosin can come and bind. So this is what gets the process started. No calcium, no contraction.
Exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge.
Okay, so with the calcium attaching to the troponin and pulling the tropomyosin off of the binding site, the myosin head can bind to this. Just a second here. The myosin head can bind to the tropomyosin. Good. And then when the tropomyosin, sorry, let's try that again. The calcium binds to the troponin, which pulls off the tropomyosin, and then these yellow blobs will allow their binding sites, which will allow for the myosin to form a cross bridge. So you got all that terminology? Calcium, troponin, tropomyosin, binding site, cross bridge, and then the myosin has a power stroke, which causes them to slide. Very confusing. So let's go through it again and do all the steps in once.
Tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge.
Okay, good. That was a good summary. And yeah, you need to know that detail. So let's look at it now in the textbook. So we can go through the textbook. So when we look at the textbook, here are our myosin. And you can tell the thick myosin because they're the ones that have the heads on them. To me, they kind of look like an aerial of paddlers in a rowing contest. So here is the myosin heads will latch on to the actin binding sites, forming a cross bridge. Then it will slide, and this will be the power stroke. So pay attention to the word cross bridge, and that's a cross bridge between the actin and the myosin. Then the myosin head bends in what's called the power stroke, and that will cause the filaments to slide past each other. So the whole sarcomere shortens, but it shortens because of the sliding. The filaments themselves do not shorten, they stay the same. But the sarcomere shortens because the fibrils or the filaments are sliding. And I know it seems like a minuscule little movement, but if you have enough of them, it ends up being quite a big movement. So in your textbook, it talks about some of the players again. So let's look at them. ATP. ATP is important because it gives the energy to activate the myosin head, which will then form the new cross bridge. Notice that and then you'll have the power stroke. You need ATP in order to break the cross bridge. And that's why when you die, you stiffen up because there isn't any energy or any ATP to break those cross bridges. So you also need calcium, and it's calcium that will pull the troponin and the tropomyosin off the binding sites on the actin. Without the binding sites available, the cross bridges won't form. So the calcium is released, it binds to troponin, and that movement exposes the attachment points or the binding sites. They attach, you get the power stroke, and you end up with the contraction. And that contraction, that grabbing and sliding of the actin and myosin will continue until there's no longer calcium, there's no longer calcium once the muscle or the nerve relaxes. Now, let's look at the complicated process of how does the nerve tell the muscle to release calcium. This is called the Neuromuscular Junction. And I'll go through the whole process in a video, but I want to introduce the players. So this is the end of the axon. The axon has what's called an axon button or an axon end. So this little lightning bolt is the action potential or the stimulus coming down the axon. Then we have vesicles, and these vesicles contain what's called the neurotransmitter. In this situation, the neurotransmitter is acetylcholine. Now, The reason we need this is the neurons do not actually physically touch the muscle. There is a very small space, and this very small space is called the synaptic cleft. So the electrical impulse of the action potential comes down the axon and will cause these neurotransmitters to be released across the synaptic cleft, bond to these receptors, and then the receptors will release the calcium. Okay, now that we know the players, let's look at the video in a little more detail.
Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system.
Okay, so this is an axon coming down, and then this is the axon terminal or the axon button.
A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction.
Okay, just see how those work together. So the signal will come down the axon, and then it will cause these yellow circles. These are the circles that contain the neurotransmitters. Then the neurotransmitters will cross the synaptic cleft and will bind to the receptors on the muscle membrane. Then if enough of them bind, it will cause what's called a depolarization, and that will result in a change of enough change in the potential to cause the calcium to be released. Okay, let's go find another video in a little more detail. This is a good video on the neuromuscular junction because it gives you a big picture.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the Neuromuscular Junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions
causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Good. So let's do a quick recap. There was a lot of detail there, but I liked the fact that it shows all the steps. So first off, the action potential will come down.
Terminal.
The action potential comes down. As it comes down, it opens calcium channels on the axon. We didn't talk about these before. These calcium channels, you'll see that calcium channels are quite often causing changes. So the calcium channels come in, they cause the synaptic vesicles to migrate to the synaptic space, and it will release the neurotransmitter, in this state, acetylcholine. Then the acetylcholine will move across the synaptic gate, the synaptic cleft, and if there's enough, cause a stimulation of the potential on the other side, and that membrane, if it's stimulated enough, will eventually travel to the sarcolemma and release calcium, release the calcium from the sarcoplasmic reticulum. Now, this action will continue until the acetylcholine or the neurotransmitter is broken down. And it's broken down in two ways. Either it diffuses away or it's broken down and recycled. So let's look at that in a little more detail again.
Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so if there's no acetylcholine there, the channels will close and there'll no longer be stimulation on the membrane, which no longer releases calcium, which no longer pulls the cord off the actin, so it can't bind with the myosin, and the myosin head contract and the filaments slide past each other. Okay, that was quite a bit, and my video is getting a little long. If that was confusing, go back and re-watch this. I'm going to continue on with the rest on a new video.